Yes, Assalamualaikum and hi. I'm Dr. Nuzul, and in this session, we are going to look into food protein. Yes, what I'm going to cover today is just going to be an overview, but if you want to learn more in depth, um, I would advise you to read more in chapter 5 of the Phenema Food Chemistry textbooks. So in this lecture, we are going to cover on the classes and structure of proteins, look into what are the chemical alterations that can happen to amino acids and protein during food processing, and what are the sources, and how can we determine quality of protein. So protein is one of the major components of food. Okay, so what makes protein unique compared to other components is that it contains nitrogen, which is around 12 to 19% of its composition. So protein biologically is being synthesized in the ribosomes, where once the messenger RNA being transcribed from the DNA, then you further translate this. So sequence of your DNA will determine what would be the sequence of your amino acids for your proteins. So once it's been translated, so now this chain of amino acids, or we call this as polypeptide, will then undergo further post-translational modification for it to become mature protein. Biologically, when it comes to functional proteins, you can further categorize them as either enzyme, which is a biological catalyst, or they can be structural proteins, contractile proteins, antibodies, storage proteins, or protective proteins. So biologically, we know the function of proteins, but in food science, okay, the function of proteins will be slightly different when it comes to our food system, because what we're going to see later on how proteins play an important role in the texture and the organoleptic properties of that particular food that give the texture, the flavor, and the aroma is actually contributed by the role of proteins. So we can further define food protein as a biologically producing proteins that are easily digestible. It has to be non-toxic, nutritionally adequate, functionally usable in food products. They need to be available in abundance and agriculturally sustainable. In terms of protein structure, there are different levels to it. It started with the building block of proteins, which are the amino acids. We have 20 amino acids in nature. And then we're going to look into the first primary structure, which is the linear arrangement of the amino acid chains. And then we have how the protein then folded into its secondary structure, and later on into its three-dimensional structure. We call this as tertiary. So this is within one polypeptide. In some cases, protein may need to go into this quaternary structure for it to be biologically functioning. Okay, so not always this the case for all proteins, but some proteins may require to have this quaternary structure. But we know that proteins, they are marginally stable. Okay, so they're also prone to protein denaturation if we shift them outside their physiological condition. So now we're going to look into the general structure of amino acid, which is a building block of protein. Okay, so if you look at this basic structure of amino acids, it has here chiral carbon. Okay, this alpha carbon here that link to the amine groups as well as the carboxyl group. Okay, so as I mentioned before, there are 20 different types of amino acids. So what differentiate one amino acid to another is the side chain, or we call this as functional group. Okay, so the chemical nature of this functional group will determine the physicochemical properties of the amino acids. Okay, because it will contribute to the net charge, the solubility, chemical reactivity, and hydrogen bonding potential of this amino acid. So we know amino acids, they are optically active and they also exist as enantiomer. Okay, so if we look in the textbooks and whatnot, normally when we write chemical structures of amino acids, it's look very flat because it's written as Lewis structure. But the arrangement of atom in amino acid in nature are actually in 3 dimension. Okay, so there are two stereoisomers of amino acids. We can design Okay, they are designated as either L or D. Example, we have either L-lysine or D-lysine. So how can we tell them apart? Okay, so it depends on the arrangement, whether it's the clockwise of corn, okay, where the CO comes from the carboxyl group of the amino acid. R is the functional group or the side chain, and then N is the amine group. Okay, so if it's clockwise, then we know that this is L amino acid, whereas if it's anti-clockwise, then it is D. In nature, L amino acid is the most abundant, and our body can only synthesize protein using L amino acids as its building block. D amino acids rarely exist naturally. Some bacteria may produce or utilize these D amino acids, but generally for the most organisms, they will need to use L amino acid. The properties of amino acids generally, if they are at around neutral pH, what's going to happen is that both the alpha, this amino and carboxyl groups are going to be ionized, and thus this molecule is going to be dipolar, or we call this as the sweater ion. So the pH at which the dipolar ion is electrically neutral, we call this as isoelectric point. But if we titrate this sweater ion with base, what's going to happen is that the amine group is going to be deprotonated. So in the alkaline condition, okay, the amine group is going to be deprotonated. But if you titrate this sweater ion with acid, the carboxyl groups of the amino acid is going to be protonated. As I mentioned, there are 20 different types of amino acids, and these amino acids, they have distinct structures and unique properties, but we can further classify them according to their whether they are charged or uncharged. This one depends on the functional groups of the amino acids, whether they have carboxylic acid, they have amine, phenol, or thiol, or that can carry charge depending on the pH. We can also further classify amino acids according to whether they are hydrophobic or hydrophilic. So amino acids may be less water-soluble depending on the polarity of their structure. For charged amino acids, they tend to be more water-soluble. Okay, so we can actually further divide amino acids into 4 main types, okay, whether they are acidic, basic, non-polar, which is hydrophobic, or polar, which is where they are hydrophilic but they are uncharged. Okay, so 2 of these amino acids are acidic, which is aspartic acid and glutamic acid. We have 3 amino acids that belong to the basic group, where the amino acids are positively charged, and then we have 8 hydrophobic amino acids and 7 hydrophilic but non-charged amino acids. So you realize that some of these amino acids has been circled. Okay, just want to show you some of these have unique properties. For example, lysine that belong to the basic group. So lysine is one of the essential amino acids. Okay, it contains this amine group with strong nucleophile. Okay, so meaning that it can react with reducing sugar during the Maillard reactions. I'm going to talk about it more in detail later. Okay, these reactions, it can be particularly strong, and what happens is going to destroy the lysine. We also have here phenylalanine, tryptophan, and tyrosine that is circled in green. Maybe I can use this. Okay, circle in green here. What you realize is they carrying this aromatic. They have this aromatic side chain that is able to absorb UV light at 280 nanometer. And lastly, on the cysteine here. Okay, cysteine is unique because it has this thiol group, also aldehyde groups in its functional group, side chain. So cysteine here, the thiol group can form a covalent bond with another thiol group. So if you have 2 cysteines in your protein structure, there's a high chance for this cysteine to form this covalent bond, or we call it as disulfide bridge. In food, however, we may also find some modified amino acids on top of the 20 existing amino acids. Examples such as this hydroxyproline that we can find in collagen, and this O-phosphoserine that we can find in milk casein. And so we know that protein is made up of polypeptide chain, which is an chain of different arrangement of amino acids. So how do they link together between these two amino acids? Okay, so we're going to look into how they can form into peptide. So here you have two amino acids. Okay, what happened is that the oxygen atom on the carboxyl group of the first amino acids will be removed together with the two hydrogen atoms from the amine group in the second amino acids. Okay, thus they will form oxygen, and now this carbonyl carbon, okay, from the first amino acids will form a peptide bond with the nitrogen on the second amino acids. Okay, so what we have here then is a dipeptide because it contains two amino acids that are linked together. Okay, so this process can be repeated in a stepwise fashion, okay, to grow the this amino acid chain. All right. Okay, so for example here, if you want to add more amino acids to the growing chain, okay, so here you have the third amino acids. The same process is going to be repeated, removal of this oxygen and hydrogen from the dipeptide and also this third amino acids, and now the peptide bond will be formed between the carbonyl and the nitrogen here. Okay, so this step will keep on repeating. Okay, but the growing chain, okay, or the growing polypeptide will always occur from the C terminus of the protein. Okay, so in this case, a tripeptide, if you want to grow them to make it into a longer polypeptide, then a new amino acids will be added in this direction. As we looked at, as we know that for primary structure of proteins is a sequence of amino acids that listed from the N-terminal amino acids. There are more than 1 billion possibility of how this amino acid is going to be arranged into protein. So that's why every protein, they have unique primary structure that will determine how protein is going to be folded into its 3D conformation. Okay, let's say for example here we have these two strands of polypeptide. Okay, 1 and 2. If you look at the composition, means that the type of amino acids for both of them are the same. Okay, they both have 2 leucines, 2 alanine, 1 valine, 1 cysteine and arginine. However, the arrangement of these amino acids for both of these polypeptides are different. Okay, why? It depends on how the genetic coding, okay, it will determine the arrangement. So even though they have the same composition, but because of the different arrangement, the protein data, how they're going to be folded will be different as well. Okay, so protein, they cannot exist as linear polypeptide. It has to be folded. This to help protein to be stable and as well later on for protein to be functional. Okay, so in secondary structure, what happened is that now this linear polypeptide will be folded. Okay, will form a bond, or we call this hydrogen bond. Okay, between the amino acids of the polypeptide chain backbone. Right, so this one doesn't involve the functional group, okay, but what happened is that the hydrogen bond can form between the oxygen atom of the carboxyl group and with the hydrogen atom from the amine group. Okay, there are two different types of secondary structure. It can either be alpha helix or beta sheet. So how do they decide which whether it's going to be folded into alpha helix or beta sheet? It depends on the binary code in the amino acid sequence. Alpha helix, if you look in the figure here, is a coiled rod-like structure and it involves only a single polypeptide chain. Alpha helix is also known as 3.6 13 alpha helix structure. What it means is that if you look at the hydrogen bond that is formed, okay, between this backbone of the polypeptide, you can see that the hydrogen of the amine group from the first amino acids is linked to the oxygen of the carboxyl group on the fourth amino acids. Okay, that's why it has 3.6, and then the number of number of atoms in between these hydrogen bonds, there are 13. Okay, so hydrogen bonds are parallel to the axis, and alpha helix is typically amphiphilic or amphipathic, means they possess both hydrophobic and hydrophilic properties. Okay, the binary code for alpha helix is polar, non-polar, polar, polar, non-polar, non-polar, polar. So we have this repeating 7 amino acid sequence. We call this as heptad. So here, just to show you a cross-section of a bovine growth hormone, which has this alpha helix structure. As you can see, okay, the amphiphilic nature of alpha helix, where one side you have hydrophobic region, okay, and on the other half is hydrophobic region. Here's the beta sheet. On the other hand, if you look here, you have this extended peptide backbone, or we call this as beta strand. Okay, to form this strong hydrogen bond between the nitrogen atom from amine group in one beta strand with the oxygen from carboxyl group on the second beta strand. You see, so this is one beta strand and this is the second beta strand. So beta strand typically has around 5 to 15 amino acids. Okay, so the binary code for beta sheet is non-polar, polar, non-polar, polar, non-polar, polar, non-polar. Okay, so the sheet can either be parallel. Okay, so if you look at this one, parallel, okay, to one another, or they can be anti-parallel. Okay, this is where the strand runs in opposite direction. Okay, so hydrogen bond, okay, they can form between different part of the single chain that is double back on itself, or we call this as intra chain bond. Okay, so it's from the same single polypeptide, or they can also form a hydrogen bond between different chain. Okay, so this is completely two different polypeptide, but they can still form a hydrogen bond, so we call this as interchain bond. See that the driving force is half the side chain. Okay, the R groups will line up above the plane, and you also have another half in the bottom layer. Okay, so normally the functional group that is in the top layer are hydrophobic in nature, whereas the one in the bottom layer is hydrophilic. Okay, so the overall structure of beta sheet is amphiphilic or amphipathic and are held together by the hydrophobicity. Contain high level of proline residues. They tend not to form alpha helix and beta sheets. Later I'm going to cover on this as well. Example given for protein that doesn't really have a defined 3D structure or secondary structure is casein. The reason why is because it contains high level of this proline, okay, residues in its amino acid sequence. Why protein can't be folded into its secondary structure if it contains proline? It's because of, if you look at proline, okay, instead of having amine group, it has this imine group. Okay, they have this ring structure, yeah, in its side chain that cause the rotation of the amine group with the chiral carbon to be not possible, so it's technically fixed. Okay, so this is the phi angle is fixed at 70 degrees Celsius, because again, another thing is this nitrogen, it doesn't have any hydrogen atom at the nitrogen atom, so it cannot form H bond. So proline is considered to be an alpha helix breaker. It can be incorporated in the beta sheet, but not in the main beta strand. It can be in the beta turn or beta band, okay, together with other amino acids such as asparagine, cysteine, arginine, glycine, and tyrosine. Okay, so now that protein can be folded into its secondary structure, okay, But still, for protein to be completely folded into its native state, it has to be folded into its 3D structure. Okay, so just now we talk about the secondary structure. It's the bond between the amine and carboxyl group of amino acids. But when it comes to tertiary structure, it involves the functional group. Okay, or the side chain. Okay, so many polypeptide, they will fold into this compact structure so that amino acid residues that is originally distant, let's say for example amino acid 1 and here you have 10, okay, they can be bring together to be closer, okay, through the folding of the protein. Okay, because of efficient packing, okay, most of the water molecules are excluded from the protein interior. So there are different interactions that can form between the side chains of the amino acids that stabilizes the tertiary structure. Okay, so because protein in, for example, aqueous solution, okay, we know that in your protein you have different arrangement of different groups of amino acids. Some can be hydrophobic, some can be hydrophilic. So the hydrophobic part doesn't like to be in contact with the aqueous solution water molecules. So what they do is they tend to stick together. Okay, they will form a bond between this different hydrophobic region, okay, forming this hydrophobic pocket. So the interior of the protein tend to be hydrophobic, whereas the exterior will be more hydrophilic, because then hydrophilic likes to be in contact with water. Okay. So there are different forces or bond that can exist to help stabilize the tertiary structure of proteins. Here we have, for example, electrostatic interaction. This is an attractive force between the positive formal charge on the polar, okay, basic amino acid residues, and the negative formal charge on the polar acidic residues. You also have hydrogen bond that can be formed between the polar side chain or with peptide groups. Okay. Here we have weaker force, okay, which is hydrophobic interactions. This is where the non-polar molecule or groups tend to cluster together in water and form this water-free pocket in the interior region of the folded and compact peptide. Okay, we also have here van der Waals interaction. This is temporary fluctuating dipole in a large methane group in amino acid side chain that can induce opposite dipole in another group of a nearby folded chain. Here, okay. So the dispersion force setup would be enough to hold the folded structure together. And this we also have here, okay, disulfide bond. So disulfide bond is also is a type of covalent bond that can be formed upon oxidation of the 2-thiol group on the cysteine. So here, for example, you have cysteine residue, okay, cysteine amino acid, because it has thiol group, they can be oxidized and form this covalent bond. So among all of these different forces or bond, disulfide bridge is the strongest. Okay, it has the highest bond energy as well, as compared to weaker interactions such as van der Waals, which is only 1 to 9, or hydrophobic interaction, which is around 4 to 12. Tertiary structure, there are two different categories. Protein can either fold it into globular structure, globular shape, or it can be fibrous. For globular protein, okay, for this globular structure, the polar residue will face the outside, okay, on the exterior. So we have this hydrophobic exterior, whereas the non-polar residues will face the interior and interact with each other through these hydrophobic interactions. We call this as hydrophobic pocket or hydrophobic interior. Example of globular protein will be enzymes, okay, or chemical signaling compounds such as hormone. One thing about globular is that their structure is not static, so they tend to be more sensitive to temperature and pH changes. So what happens if you exert higher temperature or pH, this protein will then tend to be denatured and unraveled. Fibrous protein, on the other hand, if you look at the basic shape, is long rod shape. Fibrous proteins are mechanically strong, and they mostly play structural role in nature. But because in fibrous structure, you cannot hide the hydrophobic residues, okay? Because they've been forced to be in contact with the aqueous solution, so that's why most of fibrous protein, they are insoluble in water. But in terms of its rigidity, it is more stable compared to the globular protein, and they are stable against mild changes in temperature and pH. For fibrous protein, there are two types. Either it's elastic, like gluten, or inelastic, such as collagen. For elastic, what happens is when you stretch this elastin, what you did is you didn't stretch the molecule, but what you do is you straighten the molecules in the direction of stretching. So when you remove the stretching load, this elastin molecule will restore to the original orientation and shape, become crumpled. For inelastic fibrous protein, okay, they can break if you exert more than 10% of its rest length. Okay. So protein may exist in its 3D structure as tertiary structure. Okay. Many proteins are only made up of a single continuous polypeptide chain, so we call this as monomeric. But there are also proteins that are composed of more than two or more polypeptide chain, that we call each of these chain as subunit. Okay, so let's say for example this particular protein, okay, let's hold together. Okay. It made up of 3 different polypeptide. Okay, each of these we call it as subunits. So this structure, okay, where this protein structure is held together by the same forces or interaction as the tertiary structure, we call this as quaternary structure. So it is an arrangement and position of each of the subunits in the multi-unit proteins. As I mentioned, what holds this quaternary structure together, the forces and bonds are the same as tertiary. You have your hydrogen bond, hydrophobic interaction, van der Waals, and also disulfide bridges. But the major stabilizing force for quaternary structure is actually hydrophobic interaction among the non-polar side chain of the contact region of the subunits. So in between the subunits, what hold them together is mostly the hydrophobic interactions. But it's not only limited to hydrophobic, there's also additional stabilizing force. This include interaction with the side chain of the subunit, okay, electrostatic interaction, hydrogen bond between polar groups, and disulfide bonds. Okay, one example of a protein with quaternary structure will be this hemoglobin. So hemoglobin is tetrameric because it contains 4 subunits of protein, okay, where 2 of them are alpha and 2 beta subunits. To show you how quaternary structure is being forced to be formed, so you have here folded protein. In some cases, they are unable to contain some of the hydrophobic residues. So this is the hydrophobic residues. So what happens is they are going to form a dimerization with another polypeptide, okay, folded polypeptide with exposed hydrophobic, so that it can shield this hydrophobic amino acids from water. Factors that can affect protein folding and structural stability. One is the steric constraint. Okay, so the thing is, for amino acids, okay, you have here, let's see, okay, so you have amine group and the carboxy group, right? So this, okay, this bond between this amine and carboxy group, they are flexible, they can rotate. So here between this nitrogen is pi, and then between this carboxy group and chiral carbon is psi. Okay, they can bend, they can rotate. Okay, but then because protein, they are not static, they are flexible. But what limits their movement is the bulk of the functional group or the side chain. So if you have more bulky side chains such as phenylalanine or tryptophan, this will limit the movement of this polypeptide chain. So we call this as steric constraint. Okay, another thing that's going to affect the protein folding and structural stability is the strong interchain hydrogen bond that can strengthen certain configuration. So any permanent charge on the chain can also lead to interchange, electrostatic repulsion and attraction. But then these forces will be strongly pH dependent. And lastly, what really strengthens or further stabilizes structure of protein will be the covalent disulfide bond. If I were to give an example, okay, most of the allergen, although this is not definitive features of allergens, but what we can observe for the majority of allergen, they are able to resist strong acids and enzymes as well as temperature because of the many disulfide bonds that can be found in their structure. Amino acid can also be altered. Okay, protein and amino acids can be altered due to processing. For example, hydrolysis of proteins, we also refer to this as proteolysis, is the cleave of the peptide bond. So you break down the peptide bond by the addition of water. Okay, so and then break down into smaller peptides or into amino acids. So protein, they can be hydrolyzed by acids or enzyme to give peptide and free amino acids. Okay, in food system such as soy sauce or fish sauce, this is favorable because amino acid has its own aroma or flavor. Okay, so hydrolyzed protein usually listed as ingredient on soy sauce label. Okay, so hydrolysis helps in increasing the bioavailability of amino acids. However, if you consume free amino acids in excess, it can be detrimental to your health because it increases toxicity and can also lead to liver damage. How processing can alter the amino acid is through Maillard reactions. This can happen if you exert heat to protein and you also have another precursor which is reducing sugar. Okay, we also call Maillard reactions as carbonyl amide browning or non-enzymatic browning. So this Maillard reaction, it can cause changes to the functional properties, color, and flavor of the food. From the nutritional standpoint, Maillard reaction can decrease the nutritional quality because as you can see here, okay, the chemical reactions can take place between the amine group of the free amino acids, which is lysine, and the carboxyl group of the reducing sugar, which is in this case glucose, forming this lysine and glucose complex. So once they form this, the amino acid is practically destroyed, and you can no longer utilize lysine as a building block for protein synthesis. So just to show you what happened with during the Maillard reactions, so you have sugar, okay, they form complex with the lysine, okay, forming this Schiff base, okay, by the removal of water, and this Schiff base will undergo further molecular rearrangement to form this Amadori product, have a slight color to it, and this Amadori product will then further oxidize to form this advanced glycation N product that gives it a brown color. Okay, this is the browning of the bread after baking is due to this Maillard reaction. Okay, amino acids can also be modified through alkaline reactions. One example will be the isomerization, where the L amino acids can be isomerized into D amino acids. This is not favorable because we cannot digest and utilize D amino acids. Okay, alkaline reactions can also lead to the formation of lysoalanine, where lysine become highly reactive at high pH and react with dehydroalanine to form this cross-link lysoalanine. Because lysine is a essential amino acids, if they form this complex, it's no longer become available. So this is a problem because it's a limiting amino acids in cereal grain. It can also alter proteins structure and amino acids. If it's mild heating treatment, it can lead to alteration in protein structure, and normally it's beneficial because it can increase digestibility as well as bioavailability. But if the heating is too severe, if it's more than 200 degrees Celsius, it's going to drastically reduce the protein solubility and functionality and may give decreased digestivity and bioavailability. In this extreme heat, you're going to decompose the protein. We call this pyrolysis. And degradation of cysteine due to extreme heat can lead to terrible flavor problem because this thiol group will then lead to the formation of the hydrogen sulfide. Okay, heating can also cause this amide cross-linking, which is the isopeptide bond formation. But then again, this reaction is less common because it requires very high temperature. So the normal cooking temperature may not be sufficient to cause this reaction. Here, oxidation can also alter the properties of amino acids and protein. Example given here for lipid oxidation, which is in this case the aldehyde and ketone product of lipid oxidation can react with lysine, thus making lysine unavailable. Or this oxidation can also happen to the methionine. Okay, so this will produce sulfoxide or sulfone. So normally methionine can be oxidized by hydrogen peroxide or organic peroxide. So here methionine, if it's being oxidized into methionine sulfoxide, it's not really an issue because methionine sulfoxide can still be active as essential amino acids. But if it's being further oxidized into methionine sulfone, then this methionine sulfone has little amino acid activity, so it's not very favorable. So now we're going to look into what are the sources of food protein. We've looked at the basic general structure of proteins and what are the alterations that can happen to these protein and amino acids. So here, if we talk about source of protein, traditionally we can it can come from either animal origin such as meat, okay, milk, and eggs, okay, or it can come from the plant origin sources such as legumes and cereal. But we also have non-traditional source of protein such as this mycoprotein from fungi. Okay, you may have seen this in the supermarket. This is vegan meat, okay, made up from protein of fungi. And nowadays people tend exploring potentially using insect as alternative protein. Okay, so again, with this new protein, how suitable they are to be used as food depends on the cost of production and the ability of this protein to fulfill the normal role of protein ingredient in food. Okay, so here just to show you that we can further categorize this food protein that can come from either plant-based protein, animal, or even fungi. Okay, so different proteins may have different nutritional values. Factors that contribute to these differences is depends on the essential amino acid content as well as digestibility. Okay, so we do have daily protein requirement. Okay, in our nutrition depends on the type and composition of proteins in the diet. Then further divide these amino acids into either they are essential, conditionally non-essential, or non-essential amino acids. What it means here for essential amino acids, these are the amino acids that cannot be synthesized within our body, so we must consequently supply it through the diet. Okay, conditionally essential amino acids, these can be synthesized, but the synthesis can be limited under special pathophysiological condition, okay, such as individual with severe catabolic distress or in premature infant. Okay, for these non-essential amino acids, these are the amino acids that can be synthesized in our body. Just briefly showing the recommended essential amino acid pattern for food protein. You can see infant requires more of this essential amino acid compared to the adult. Okay, so here you have this random advertisement that you can find in internet where Dr. Joe Furman give you this comparison. So between these two, steak and broccoli, if you can see from this ad, is that broccoli has higher amount of protein. Okay, and then steak. What does this tell you? Does it tell you that broccoli is better than steak in terms of its protein value? Okay, can you trust this advertisement? Okay, so in terms of quality, do you think broccoli has better quality than steak? Well, it's not. I'll tell you why. So before that, we're just going to look into proteins. Okay, in food, it's not always only rely on the composition. We also want to look at the quality of proteins. So high quality proteins these are proteins that contain all the essential amino acids at levels greater than the FAO, WHO, UNU reference level, and it has digestibility comparable to or better than egg white and milk proteins. So animal proteins, they are of better quality than plant protein. Okay, so why? As I mentioned before, high quality proteins mean that it contain all the essential amino acids. So here, just to give you a chart of different food, okay, different food source and their essential amino acid content. So as you can see, for animal proteins such as lean beef, cows, milk, and whole egg, they all contain the essential amino acids above the reference protein here, or at least on par. Okay. But if you look at plant-based protein source such as wheat flour, maize, soy flour, bean, and potatoes, they may have limiting amino acids where the amount of these essential amino acids is lower than the reference protein level. Okay, for example, wheat and cereal, they tend to lack in lysine, whereas soybean and other legumes, okay, they may be lacking in methionine and cysteine. Okay, so if you are vegetarian, you have to make sure that you diversify your diet. Okay, you have to eat legumes and grain and cereal together so that you can get sufficient amount of essential amino acids. Composition of essential amino acid alone is not enough. Another factor that determines the protein quality is its digestibility. So true quality of a protein depends on the extent of which these amino acids are utilized in the body. So the more proteins are able to be digested, the more these amino acids will be available, okay, for absorption. So proteins of animal origins, they tend to be more completely digested compared to plant. Okay, this is because there are a few factors that can affect digestibility, such as the protein conformation itself, okay, the anti-nutritional factors that you can find in legumes, and as well as processing. So protein conformation for the fibrous protein is less easily digestible compared to globular proteins. Okay, for the second point here, these anti-nutritional factors, some of the legumes, this is one of the reasons why you can't eat raw soybean or raw cereal, is because the kernel, okay, or the shell of these beans or the cereal, it contains high level of these anti-nutritional factors. So these anti-nutritional factors can inhibit the absorption of amino acids in your intestinal lumen. Okay, some of these can be removed through washing and cooking. Okay, some may remain, but these are one of the factors that will reduce the digestibility of proteins. Processing is a mildly denatured protein is more easily digestible compared to the raw proteins.